Dr. Anderson has more than 110 peer-reviewed articles and holds 5 patents. She was a member of the Gulf Research Program Advisory Council for the National Academies of Science and has served on the board of directors for both North America and World Council for the Society of Environmental Toxicology and Chemistry. Dr. Anderson has been developing passive samplers since 1999 and in 2008 developed the personal silicone wristband sampler technology to measure individual chemical exposure. So we are thrilled that she's here today to give a presentation on the development and use of silicone wristbands as an exposure tool. So thank you, Dr. Anderson, and I'll hand it to you.
Thank you. Just verifying that you can see my slides. Can you see my slides?
Yes, actually we see the notes view.
Okay, just a sec.
So I think if you go to display settings, you just say swap presenter view.
How's that?
Perfect.
Hey, Will. So thank you for the introduction and thank you for the invitation to come speak. Remind me again how much time I have. I tend to jabber on.
25 minutes, but we can be a bit lenient.
Don't tell an Italian woman that. Today I'd just like to share a little bit at a high level about some of the things we've been doing with silicone wristband and general passive sampling, some background about the technique itself, the application, and how we've applied it. So I think it'll overlap really well with some of the presentations this morning. I don't doubt I have to tell folks here that sometimes stationary monitors are a poor estimate for personal chemical exposure. And that's really where my interest stemmed from. I've made many different types of stationary monitors for fit for purpose of understanding bioavailability and different chemical classes. But it was really the connection between stationary monitors and the individual exposure that got me interested in trying to design something that could be worn by an individual, because there is kind of this unknown at the time, but we highly suspected that stationary monitors might not be a good estimate. So we started looking at personal samplers, and I've worked in the passive sampling with different kinds of polymer, carbon polymers and silicone polymers, and we eventually ended up on the wristband. Again, trying to understand the environment and the chemicals in the environment as an important contributor to disease in humans. So sort of where my interests lie in trying to understand developing this technology. So with that, we tried to build a sampler with purpose, not to solve all problems, but to solve a slice, and one of those being quantifying bioavailability. From my work in environmental tox and environmental chemistry, the use of passive sampling, different types of carbon polymers and silicone polymers, was to try to capture from the environment the bioavailable fraction, whether that be in waters or sediments or the air, was trying to understand the difference between what is there and what is biologically available from the exposure side. So many people before me, obviously we stand on the shoulders of many, have developed passive samplers to try to take up that space and try to develop different kinds of polymers that would, very roughly mimic biology. And so that's sort of where my history came from. And I brought that history from the environmental side into trying to develop something for that people could wear. and we eventually expanded that beyond people, but companion animals and so forth. So the passive sampler has both lipophilic, meaning fat-loving nature, and pore sizes that are quite close to a cell membrane. And so just trying to bridge that gap a little bit between what's in the environment and what is actually an exposure. So as we developed the passive sampler in the 2000s, late 2000s, and then we eventually published that, and that sort of started the ball rolling with the silicone wristband, and in general the approach of putting these passive samplers on folks. So the idea also was fit for purpose. So build a passive sampler or build a sampler that people could wear. We want to actually have a lot of the questions that you all will probably ask me at some point this afternoon about how do we know this, how do we know that. We wanted to really do all of that as much as possible leading up to our publication. A lot of times different kinds of chemicals are not always tested in the matrix they're collected. Like, for instance, how long can you keep chemical X in urine at temperature X, right? If there's someone had to do those work. When you build a brand new technology, in this case, a silicone wristband, we had to do that. So we had to do, what is the testing that's necessary, which is often called storage, transport and storage stability. So we tried to do all that because it's important to understand the application of the technology. First and foremost, what we wanted to do was make something that was connected to the bioavailability, right? We wanted to get that fraction from the environment, and we wanted it to be on the individual. We already have great monitors for doing stationary monitoring and lots of choices there. What we wanted to do is get something on the individual that was easy to use, didn't require power. It's one of the downsides of some of the technology. So we wanted it to be power-free and something that would be easy to wear during all of life. So whether that's, sleeping or doing your everyday activities. And so those were some of the reasons why, there are goals of the passive sampler. This is one of our first studies on transport and storage stability. We have subsequently done more, but the first study out was 148 chemicals. And we looked at transporting those samples at various temperatures, storage at various temperatures, and what was their stability. And in the end, the short story, you don't have to go into all of these different graphs, but the short story is we can transport at ambient temperature, which was another really important goal of the passive sampling technology, because it is really hard in remote areas, or even not in remote areas, but during times of stress or disaster, to have to deal with complicated transport conditions like -80, -20. And so really, I think that was an important component of the design of the passive samplers, was that we'd get something that was easy to transport. So we've subsequently tested these for many years now. So another attribute was to be able to archive these samples either as the wristband itself or as the extract. And we've proven that both of those things, when stored at -20, have really long storage stability studies. This was just the first one, which was 6 months. We've subsequently done other studies extending these out to multiple years. So that was an important component. We also had questions, sort of left field questions like, well, you have a sampler, but your wrist is in the sun and that might degrade something like pHs. So there was a lot of pushback. Oh, there's going to be UV degradation. Turns out that the wristband actually stabilizes these chemicals in ways that we could have predicted. But having the data is certainly important. So we actually stuck the wristbands on top and beat the heck out of them with some UV. And I know it's hard to believe that the sun does come out in Oregon for those in Southern California, but it does. And so, we did that study and showed that there were no statistical differences with the pHs. They were quite stable. even if we left them out for days in the sun on reflective surfaces. So the other attribute that we wanted to incorporate ideally was to be able to look at a lot of chemicals. One of the things we hear back is that there's a type of sampler that's used and it can only measure one thing. That gets to be difficult for those to do study designs because it's so limiting. So it's like, well, can't put 20 samplers on an individual, or we can't afford, probably just as important, we can't afford to put 20 samplers on individuals, but yet we have this interest in a really broad range of chemicals. So one of the attributes of the passive sampling, whether it be a carbon polymer, in this case the silicone, is that it's applicable to a wide range of chemicals. And I can throw up different kinds of chemical physical properties, and a lot of times it's hard for people to get, it's even hard for me to get a feel for that. What does that mean? Boiling point, or log KOA, in this case, octanol air partition. So I just put up as an example, in the case of log octanol water partition, we have demonstrated that we can measure chemicals in the wristband over 8 log units. Well, what does that mean if we can measure chemicals over 8 log units? How different are those chemicals? So I just kind of use the analogy of temperature, water. In this case, 0 degrees to the temperature at the center of the sun would be 7 log units. And this is a very educated audience, so you guys don't need that, but I still think it kind of gives you a sense of how wide the chemistry range is that is applicable, that we've demonstrated with the wristband. So then it's a question of doing all these things inside the laboratory, but then the real rubber meets the road when you actually do studies. And so we have now done thousands of wristbands in various places where we've tested with an actual biomonitor, with other types of traditional sampling technology in various kinds of communities. And these are just some pictures of various places we've been. I'm glad to answer questions on any of them. They're all on my website, which I'll identify at the end. And I'm glad to talk about them, but just to kind of give you a sense of the wide applicability and the quick adoption. I mean, new technology usually takes decades to get integrated. It's not like sort of computers, which are really quickly integrated. Chemistry technology takes a long time. You have to get everyone on board with how to do the chemistry part and be ready to adopt a new approach in the field. And a lot of times there's resistance to new versus the comfortable. And so really in the last 10 years, it's been pretty amazing how fast this has come along, in my view. I guess I was expecting slower. So I'll talk about a few of these studies, and again, anyone is welcome during the Q&A to ask me anything as we go on. So the very first study that we did was with roofers, so an occupational exposure. And so we had roofers work days, and then we had roofers at a training site. And I guess this really stuck to home for me, just how important the individual wristband was for understanding individual exposures. So one of the outcomes of the study was, interestingly enough, the training site in Oregon had higher pH in the wristbands of those trainees for 40 hours and 8 hours than the actual roofers did. And so that was kind of interesting and counterintuitive, because they're training and they're in this, big facility versus, thinking of a more intimate, right on top of someone's roof, going, such as this. And so it was kind of surprising. We subsequently learned that in Oregon, the training site, because it does rain here a lot, that the training site is roofed. And that roof actually holds the tar fumes in clearly. You have 4 times higher pHs in the wristbands than the outdoor workers. So whereas the outdoor workers, you have this sort of open air component, versus the training site has a roof on it so that they don't have to get rained on while they're doing training, but that holds in, even though the sides are open. Not a lot of, there's less movement of the air. We've subsequently looked at military. I'm not going to share that data. I'd be glad to talk about it. So one of the other components with using technology is to get adoption by communities. And in all cases, we've had really good enthusiasm about using, almost too much enthusiasm about using passive samplers. We've looked at children as young as 3 and community members as old as 93. And so there has been good adoption, and we've actually discovered a lot of things about whether it be flame retardants, and turns out children do have a lot more flame retardants than adults because of their nature. You might of the activities, and again, you wouldn't be able to necessarily pick that up if you had a stationary monitor, and of course a lot of complications pulling blood samples from that kind of a population study. This is a study that was done about a decade ago, and or 7-8 years ago. And this was where we had a very small, tight community in Ohio, and we had stationary monitors all over the place. But then we also had community members wearing wristbands. It's kind of a classic example now of where we've shown that the stationary monitors are not really a great reflection of the individuals. And so in this case, our stationary monitors, whether there was a natural gas fracking well on the property, much higher, just as you expect. Those that have one sort of adjacent as a neighbor, and then none. So kind of a typical stationary monitor. We had concentric circles around various places. And what we found, though, with the wristbands on the graph on the right is that well, there is an association here clearly between the stationary monitors and the wristbands. You see that there is, in fact, some underestimates and overestimates on the individuals, and this is a pretty small study. So, and each have their own, both underestimating and overestimating risk are equally bad. And so an example of where knowing these individual exposures are important. And this is just PAHs and in a very, very small community, as an example. The use of the wristbands, we have found, have been really nicely accepted by many communities. We did a study, I think it was in 2015. We had been in this Senegal community for about a decade. And so we had done all these exchanges back and forth. And so these folks knew about the wristband because we've been working on them in the laboratory, and they wanted to be one of the first to use the wristbands, which is very sweet and nice, because they're wonderful group to work with. It's a very small community, and a lot of the folks in the community have similar jobs, and we actually also had people from the same household in the community. So it's a small study, one of the first ones that we did. And we had folks in the community wear a wristband 2 different periods, 5 days each. And what was interesting, and of course all of the adjacent types of questions and so forth. And so we had 35 folks, and what you can see on the graph on the left is that all 35 of them are different. So even though we have people with the exact same jobs working in the same fields even, and we have some people who are in the same households but different jobs, all 35 were different. So it's kind of another example too of understanding the exposures at a community level really are individual, and I guess viva la difference, right? I mean, I would like to think that I'm individual, and then that's a good thing and to be celebrated. But then when we're trying to understand exposures and chemistries of exposures, then that makes it harder. But the wristband certainly can help. The other part of this little study was that we had them wear them for 2 different periods of time, and we found that within a given period of time that you were very much like yourself. While you were different than the other person that lived in your home, or you were different than the other person that worked nearby you in another agricultural field, you yourself were very much like yourself week to week. And that sort of made sense after we had the data, because while my husband and I have we share the same household, he starts, he's goes to a different job than I do, but I start off my morning making my coffee, and I come to campus and I drink my coffee and meet some people, and then I go take my dogs for a walk, and then I make some dinner, and that whether that's a Monday one week or a Monday another week, that's very similar. And so what that tells me is that we tend to have the same exposures. Our life is kind of has some similarities, but we're very different even with other people in our communities. I guess in hindsight, maybe that was a duh for a lot of people, but I found it quite interesting that here we are with the chemistry, and that's what that is saying. And so from that study, we detected 26 different pesticides, and you can see again, going to chem phys properties, there was a really wide range of pesticides in folks' wristbands, everything from dimethoate to bifenthrin. We saw anywhere between 2 and 10 pesticides. And from the questionnaires, all pesticides reported by the participants that they had used or were in fields that had been used in their fields that they were working in were found. That's a good thing. But what was the companion to that were that we found 19 pesticides in their wristbands that hadn't been reported by participants. So the good of questionnaires and I guess the bad of questionnaires all in one little baby study. So understanding chemical exposures, I think in an ideal world, we'd have biomarkers for every chemical, but we don't live yet in that ideal world, and we're a long, long ways from that world. So all chemicals do not, in fact, have a clear link to an internal biomarker. So while that might be a gold standard, it's not a current standard that we can achieve. But we still want to understand our chemical exposures, and we certainly want to try to do interventions where possible when we do understand what those exposures are. And those interventions are not going to be off of a biomarker either. Those policies and regulations are going to be based on some kind of chemical exposure on the external exposure. You're not going to see a regulation based on a biomarker in a person's blood for all the reasons that I list. So there's space. It doesn't mean that biomarkers don't have super important space in understanding chemical exposures and health effects, but there's still space for understanding that external exposure, which is the space that the chemical silicone wristband sets. Small study, again, one of our early studies looking at DNA damage and pesticides in wristbands, and we did find an association, but in children in North Carolina. So I think you saw an awful lot of data on pHs earlier, so this just kind of reiterates some of that data. We have urine data with polyurethane foam samplers, urine metabolites with polyurethane foam, whether filtered or unfiltered. And then we have the urine metabolites with the wristbands. And what we found in this study was that the wristbands did seem to have more positive significant correlations between the urine pH and the wristband than the puffs with or without filters. So there's a place, I just personally like to have more tools in my shop than fewer tools, and then I pick the right tool for the job. So there's certainly a place for the backpack puff, but then there's also a place for the wristband. So I guess I collect tools. So this seems to be one that would have value, and hopefully you folks can see some of those things. So how do the role of silicone wristbands and biomonitoring? So we have now expanded our studies with children, as I mentioned, every age group from 3 to 93. We've also put the silicone wristbands on cats and we've looked at hypothyroid, both flame retardants and other. We have a screen with about 300 endocrine disrupting chemicals. And so one of the nice things about the silicone samplers is their ability to archive. So we have sample archived, and we can go back and now look at, as we develop additional methods for looking at the wristband, we can now use those methods to determine other additional hypotheses. Just kind of trying to wrap up here in the next few minutes. We've applied these to many different places. We look, as I mentioned, a whole host of types of chemicals, from different chemical classes to different uses of chemicals like personal care products. And we've had done everything from the Arctic to Antarctic. In this study, which has only about 250 people, but from all over, we see the same kind of thing we saw in Africa, which is that no two individuals had the same chemical detection profile. With that said, we did see that there were many chemicals that were common. So, whether it's a phthalate where we have 90% detected in all those different places, or a fragrance compound, we do see some similar chemicals at very high frequency rates in these wristbands, some of which have had little study. And I think that's an interesting component that we can bring with the technology. Finally, now, since I'm going through this myself, the role of the wristband in biomonitoring in environmental disasters, where this is wildfire disasters, first responders. We have had many firefighter studies and structural firefighters now with wristbands, as well as community members. We've had the wristbands in train derailments, cleanup crews, citizen science focus projects, flooding, hurricanes. So quite a few different ways. And I think part of that whole piece of being ready for a disaster is it lends itself well to the wristband, because there isn't a lot of like, oh, we have to ship this on dry ice, right? That's not going to happen when there's no power in Houston for a week due to Hurricane Harvey. And also the breadth of chemicals. I mean, I quoted the Houston Health Department saying there are millions of contaminants. Maybe yes, maybe no. But what you can for sure say is that in any given disaster in a complex megacity like Houston, is that you won't know ahead of time what are going to be the important chemicals that you should be looking for. And a technology that can grab lots of different chemical classes is going to have value. I think that's part of what we can bring. So with that, just sort of wrapping up, what are the limitations? I haven't listed all of them, but they are on my frequently asked question page. A few of them that I'll go through. These are organic chemicals. They're not PM 2.5. We clean the particulates off them. They're not formed metals, so we're not going to find chrome 6. They are time integrated. They're not real time. You do have to take them back to the lab. So one of the questions I used to get, do they change colors? They do not change colors, but they do have to go back to the laboratory. Where are we with that turnaround time to real time? Right now we have a purge and trap method for VOCs for the wristbands. And so from the time you drop the wristband off to when I can give you a result, if I'm already got the purge and trap running, I can give you a result the next hour. That's not real time. In a 2-week period, we actually analyzed 1,000 wristbands. Not real time, but in my world, that's pretty good. We have worn the wristband for as little as 8 minutes in a firefighter demonstration piece, and we were able to detect PAHs. However, in that that's a contrived situation. I would say that the samplers should be worn for a few hours. I like 7 days just because I get a work day and a weekend to understand your total exposures, but they could be worn for a month. They could be worn for multiple months. They are an external exposure obviously, and an independent measure. And I'll probably stop there. There's a lot of limitations. Don't try to hide from them. Again, I just feel like it's a tool that that as someone who's interested in exposure science would want to learn about. I will point out that my management plan over here on the far left requires that I give a disclosure on my acknowledgement pages. I realize from your folks that normally you would give that on the front page, but I was not able to get through my management plan. And I'm sure they would have approved it, but I'd rather stick with my management plan than the SOP. So here's my acknowledgement that I have a conflict of interest that OSU manages. And lots of people to thank, too many to go through. But at this point, I think I'm at my 30 minutes, so I will stop there and answer any questions. Or wait till the Q&A period.
Thank you so much, Kim. So we'll take 15 minutes for questions now, and we'll have a longer discussion period to talk more with Dr. Anderson after the next presentation. But let's start with any questions now for 15 minutes. Anyone has one? Yeah. Go ahead, Jenny.
Hi, Kim. Thank you for that. Jenny Quintana at San Diego State University. You've talked a lot about what a great tool that wristbands are. Mostly you're presenting worn wristbands, as well as the pet one with the little hanging pet tag. But recently we've been hanging them in the air, mostly for third-hand smoke or tobacco-related issues, but also for other air pollutants in environmental justice communities. And I'm just wondering if you could just briefly comment on the utility, just hanging them up on a outside the environment.
I get that question a lot. Thank you for asking. So we have put them on cats, dogs, and horses so far. So just to kind of finish that thought. I'm not a big fan actually of hanging them in the environment or hanging them on a tree or something. And the reason is most folks would like to calculate an air concentration. And right now, I don't have all of those calculations ready to give you. We're working on them in the lab right now. So you're going to be able to tell what is in the wristband, but you're not going to be able to calculate that back to an air concentration. So if you were interested in pesticides or PAHs, there are other samplers available to you that you would be able to calculate an air concentration back to. So I usually like to direct people to those. 3M has some, there's other people. I mean, we use other passive samplers so that we can, again, get that air concentration. So I'll just stop there and you can have a follow-up or not.
Say thank you for that. Yeah, I think that if one's looking for the presence or absence of compounds, such as with non-targeted, sometimes they can have a different pattern, So that's interesting. And it's amazing to me, even semi-quantitatively, not worrying about what the actual air concentration is, but comparing, let's say, indoor environments to each other, like you can actually relate that to behavior in the home. Thank you.
Yeah, so you can do that also with the other samplers. We do semi-targeted, and then a colleague of ours does non-targeted. You could do that with those other samplers too. So you're not relegated to those quant methods, but yeah, you wouldn't necessarily calculate back from a non-targeted method to an air concentration. Still kind of feel like there's more known about those other samplers than the wristband as far as an air sampler. And not every commercial laboratory knows how to deal with the silicone sampler. So just that's sort of another caveat. I saw another hand. Go ahead.
Oliver has his hand up. Oliver, do you want to ask a question?
Thank you very much for your presentation, Dr. Anderson. This is really enlightening, and it's I feel your pain when you said you have to, or joy, when you say you had to validate every single question yourself, and the duration and the storage and the the do's and the don'ts, and that's great. Now, it's been a couple of years now that these, many years, I'd say, that the wristbands are out, and I wonder what's the adoption in the field, because of course, the more studies are there on wristbands, the more people will like it and apply it. But is there anything similar to NHANES or other large-scale studies where they almost become authoritative, where at some point people say, yeah, you gotta have it. You can't just rely on, say, urine and whatnot, but you have to have the wristbands. What is your take on the adoption in the field?
Yeah, so I think there's about 150 publications, only like 20 or 30 are mine. So I would say from an academic standpoint, there's good adoption because it was a pretty slow rollout. I mean, I was by myself essentially in the first few years, sort of, talking about these and probably talking about them in communities that weren't necessarily the early adopters, as far as I wasn't going to the ISIS meeting, initially. So I still think it's being accepted by the academic community really quickly. The military seems very keen on adoption, so I think that's interesting. You bring up something very near and dear to my heart, but I have not been able to break through, which is NHANES. I think you are absolutely right that if it were put into the NHANES, and it's perfect for NHANES because they end up archiving so much and not necessarily doing all the testing on everything they collect, and this particular technology archives really well for future interest. I think that's right. I would like to get this in NHANES as a technology, and then I think, yeah, then you've made it to the big times, as it were. That process, I keep pinging through that process, and there's some bureaucracy there, and that's probably not my best gig, but I just agree. I agree 100% with you. When you make it to being in something like NHANES, then that's just it. Not sure how to complete that circle, but I feel like the more people who use it, like I said, there's about 150 pubs, and I'm really surprised how many are coming from all different places on the planet. That it is making lots of headway in certain space. And there's a certain amount of, it's not ownership, but it's like, I really want to emphasize using it in its fit for purpose, because if it's not used in its fit for purpose and people misuse it, then that's going to give it a bad rap, but that's because they're not really using it for its, in the way it was intended. So I do like to get out and say, like, this is where this is the really good lane that it's in. Be careful over here, because then, it's not really what it was intended for. So there is probably too much passion on my part to make sure it gets used properly. But yeah, I totally agree with you.
Let me check if there's any questions that have come in online, Rebecca.
We do have a question from Jeff Wagner. Jeff, would you like to ask your question?
Sure, thank you. This is Jeff Wagner from CDPH. I was curious if you've done work with field replicates, say one bracelet on each wrist, maybe from the same individual, and looked at like CVs or even common chemicals detected or not detected.
Yeah, so actually in the very first study we did with roofers, we put it on both wristbands. And you see a difference in roofers because they preferentially use their dominant hand versus their less dominant. And it didn't really matter for the certain work type. So depending on the roofer's job, if the roofer was, I forget all the names of the roofers now, their job titles, but if they were like pushing the stuff on the roof, there wasn't really any difference, but the guy who was, and it was a gentleman, the guy who tended the pot where all the creosote was, his dominant hand, which was closer into the pot, had higher pHs than his non-dominant hand. Yes, we do it all the time internally and in studies where people wear 3, 4, 5 wristbands. It's a pretty common thing for us in all of these studies that we load up each other with a lot of wristbands as we're doing these things. Probably 50% of all the samples I run in my laboratory are quality control. I'm actually a good laboratory practices facility, gone through multiple audits, both for EPA and FEMA and private. So, and I actually have taught classes on GLP back in the day when I did a lot of pesticide re-registration. So we're pretty heavy, probably too much so, on quality assurance. So there's an everything in my lab is pretty heavy in the quality assurance. I've had a quality assurance program plan. Like I said, I brought it with me in 1999. So I spent 10 years in a QA lab prior to coming here. So lots of replicates, lots of overspikes of every kind, lots of labeled compounds for recoveries, both surrogates, internals, and other QC samples throughout the process.
That's great information. Thank you.
Field trips, trip blanks, both. Every study, every time.
Did you want to? Okay, let me take one more question, I think, and then we'll move to the next speaker.
Hello, Kathleen Atfield from CDPH, Biomonitoring, California. So I want to press you on your earlier point. What advice would you give to our program if we were to use the wristbands as far as avoiding the missteps? So avoiding the ways that you could use these and maybe not learn what you think you're learning from them.
Yeah, I think that lots of easy things to do. The recruitment is easy, the consenting is easy. Getting people to wear them is usually pretty easy. I think we did have one 3-year-old who kept biting the wristband, but assuming that you have something 3 or older, I think until we release this paper we're working on for what's called performance reference compounds, I would design the study that everyone wears them the same amount of time. I think that's an important component until we have our PRCs worked out so that you can compare so you can compare bifenthrin with participant 1 and participant 351. So bifenthrin. A lot of the things are the interpretation. So again, until we get the PRCs, I think that performance reference compounds, those are usually labeled compounds or non-natives. That's an important component is the study design set up so that you have everyone wearing it nominally the same amount of time. As I mentioned, I like to have people wear it workday and weekend because I think a lot of people's weekends look different from an exposure standpoint than their weekdays. I've done a lot of studies with indoor, outdoor samplers and the wristbands. That's always interesting. Takeaway indoor air is terrible, which you all probably know more than most audiences. I think that's the biggest, and then recognizing what you can't do. So, you can't do PM 2.5, can't do metals. I think the length of deployment is important and the continuity of that is important. That's probably one of the things I always try to stress when people use the wristband.
And this is Stephanie John. I just have a really quick follow-up. In terms of comparing similar time points, how exact do you think they need to be? Could you compare a 24-hour sample with a 36-hour sample, or does it need to be plus or minus a few hours?
Yeah, so that one's probably harder on the chemicals that are more quickly taken up by the wristband. So if you were looking at 1,3-trimethylbenzene, that one might be harder to compare a 24-hour with the 36-hour. But if you were looking at phenanthrene or a flame retardant, probably not so much because those are pretty similar times. But the quicker the analyte comes to equilibrium, the more important it is that the times be closer together. So if you're looking for pHs, so that's naphthalene and above, the difference between 6 days and 12 hours and 7 days, not so much of a difference. Is what we've seen and even then through our mock calculations. But that would be different if it were 24 hours and 12 hours with toluene. There would be a difference probably. Does that answer your question?
Yes, thank you.
If not, then it should be an email. Anyone have any follow-up questions, you're welcome to email me.
Fantastic. Thank you, Kim. Okay, I think we're at time, so we should move to our next speaker. But we have Kim for the discussion section as well. Is that right? Are you able to stay on for the?
Yeah, I mean, right now I'm under a mandatory evacuation and there are.
Oh dear.
Yeah, thank you. For wildfire, and they're saying they might let us back in because we went from code 1 to code 3. We skipped code 2. So we had to get out within 10 minutes and I didn't have everything packed. So yeah, it's okay. Our house is still standing and they're making good progress. But if I do get a notice that they open the road so we can go, I might leave, but I'm not optimistic that's gonna happen. But I don't want to be rude. But I do have my great grandmother's dining table that I would really like to get out if I can.
Okay, well, we're hoping that does happen then. Sorry to hear about the evacuation. Okay, so our next presentation is from Heather Stapleton. Dr. Stapleton is an environmental chemist and exposure scientist in the Nicholas School of the Environment at Duke University. And her research interests focus on the identification of halogenated and organophosphate chemicals in building materials, furnishings, and consumer products, and estimation of human exposure, particularly in vulnerable populations such as pregnant women, children, and firefighters. She currently serves as the director for the Duke Superfund Research Center and director of the North Carolina Firefighter Cancer Cohort Study. Today, she will give a presentation on the strengths and limitations of silicone wristbands in assessing personal chemical exposure. Welcome.
Great, thank you. Can you hear me okay?
Yes, we can hear you great.
I will bring up my slides here. These more. Can you confirm that you can see them, or is it this notes section?
We see your slides.
Just fine. Okay, great. Well, thank you for that introduction, and I'm happy to be here to talk about some of the work that we've been doing in our lab using silicone wristbands. It's really follow up to the great work that Kim has done and just explained to you in the previous presentation. So conflicts of interest, I really have no major conflicts of interest. I will just state that I am a science advisor for the San Francisco Estuary Institute on their emerging contaminants work group, and all of my funding sources are listed here. Right. So one of the reasons I'm really interested in using the wristbands is because I'm really interested in exposure science, but particularly this concept of the exposome, right? Which the exposome, as defined, seeks to assess the totality of exposures over the life course and understand how these combined stressors impact health. But the exposome is very, very complicated, as you can see from this diagram from Roel Vermeulen's paper in 2020. It's multifaceted. It's not just chemical exposures. It's social, lifestyle, physical, chemical factors, etc. But it's this concept of trying to understand exposure over time that's really interesting to me, and how it applies or how we can use wearables to support this. Because a lot of prospective epi studies, you're getting maybe a biological sample once a year, once every 2 years, but our exposures change quite a bit over time. And I think wearables have an ability to help us understand exposure over time and patterns of exposure, particularly as it relates to our behaviors, our built environment, consumer products, our occupations, etc. And as I know Dr. Anderson already explained, silicone wristbands have their strengths, they have some limitations. I think based on the state of the science, the data suggests that they are capturing chemicals in which exposures are occurring via inhalation and through dermal exposure. And then I have this question about whether it's capturing chemicals that maybe the exposure route is inadvertent dust ingestion, particularly in children. I think we have some data suggesting that is possible, but I think that needs more work for sure. And obviously, we're not capturing any kind of dietary exposure. That could be a strength or it could be a limitation. It really depends on your question. Right. And we know that in general, chemicals accumulate initially in a linear fashion, and then eventually they reach this equilibrium portion. We do try in our studies to keep within that linear uptake phase. And in our case, we do normalize to deployment periods. What's great about them is that they can integrate your exposure in all the different microenvironments that you spend time, if that is your question, right? Because we know our exposures are very different at home versus maybe commuting in your car or on the subway or in your work environment. So what's great is they go everywhere with you, and then they're integrating that exposure that you're receiving in all those different microenvironments. But maybe your question is really what's just your exposure in your occupational setting? And then they have utility in that application as well, because you can just have someone wear them just while they're working to isolate that exposure, which can be difficult by relying on biological samples because you can't disentangle what portion of the measurement internally is from your diet versus the ambient environment. So what I hope to do in the next 20, 25 minutes here is kind of go over some of the questions that we've gotten a lot in terms of how well do they correlate with internal dose? How long do you wear them? Can children wear them? I know that Dr. Anderson covered some of these already really well. Is there variability around the wristband? I know that question came up. We have some data on that. And then I was going to talk about one of our studies with wristbands, which is with firefighters, kind of showing you how we've been using them, what type of questions we're asking and trying to answer. And then I'll just close with some strengths and limitations. So let me just start with how we are using the wristbands in our lab. This is a picture of our wristband kit. We have these Mylar bags that are resealable. It has a label on it for the participant's ID. And then we ask participants to record the date and time they put it on and the date and time they take it off. And we also send them a YouTube video that shows them how they should be wearing it and recording this information. It's a really quick 3-minute video. Inside this bag is an aluminum tin with a screw top lid, and the wristband is inside. So when they're done wearing it, they put it back in the tin, they put it back in the Mylar bag. They either give it to one of our study managers or they mail it back. Sometimes we provide self-addressed envelopes. We get them in the lab and we store them in the freezer until we analyze them. And we are only analyzing a very small part of the wristband. It's about 0.7 grams, 1/5 of the wristband, so plenty to archive. I know this is a complicated slide, but this is kind of an overview of the approach we're using. We do buy these in bulk and we clean them. Historically, we were cleaning them with the solvent extraction and the sock slits, but we have moved to a heat cleaning method where it just gets us away from solvents, hazardous solvents, and allows us to do more at one time. So they're cleaned under high heat, under vacuum with nitrogen purging to kind of remove particularly siloxane residuals that are in those wristbands. And then we keep them in airtight containers until they're deployed. We store them in the freezer after collection. And then for analysis, we do have different panels that we use depending on what the study is and what the target chemicals are of interest. Most of our panels use GC, high resolution mass spectrometry. We have 3 Orbitrap systems that are operated in different modes to be sensitive to different chemicals. What we do is we take that small piece of wristband and we extract it using sonication, and then we do conduct a dispersive fluorosil cleanup to remove some lipids, things like squalene that sometimes get on the wristbands to purify that extract a little bit. But then we take that extract and we can inject it into all 3 of these mass spectrometers to collect a wide range of data. We have our workhorse on the left, which is our Q-Exactive that's operated in electron impact mode. And we perform both a targeted and non-targeted analysis in the same run with the same method. So we run calibration standards for all 113 chemicals that you can see here on the left, which span flame retardants to pesticides to PAHs, combustion byproducts, industrial chemicals, etc. And we support non-targeted at the same time, and I'll mention that briefly. But then we can take that extract and run it on our Exactive system, which is set up in negative chemical ionization mode that allows us to get these high molecular weight BFRs, which can be difficult to measure unless you have a very short, thin column with a specific type of inlet, makes it more sensitive. And we also measure dioxins and furans in that method. And then we can take that extract and also run it in we have an Exploris Orbitrap, which is set up in positive chemical ionization mode, which is more sensitive for some of these volatile PFAS that are of interest for some of our studies. So that's a wide panel. It collects data about 170 different chemicals in total. But sometimes we have interest in measuring non-volatile PFAS like PFOA, PFAS, or parabens, triclosan. In that case, we will take a separate piece of the wristband, extract it in methanol, and analyze it via an Agilent triple quad LCMS using isotope dilution. Now, I just mentioned we do have this non-targeted method we also apply. This was developed by my former postdoc, Nick Herkert. When we run these samples for both target and non-target, it collects a full spectrum, all the features or chemicals that are identified. So we use our vendor software to pull that data out. We use their deconvolution program that also does alignment of the peaks and library matching. We have 3 libraries for mass spectral matching, the NIST mass spectral library, Thermo Fisher's high resolution library, and an in-house library that we've been curating. That data is then exported with a custom R script that we developed to do some standard QA/QC procedures, mainly blank subtraction and normalization to internal standards. And then we pull in data from online resources, particularly PubChem, CPCAT, and that data is used in our algorithm to rank these features and their annotations as likely matches. And so then we can develop a list of annotations for each feature in our workflow. And then we visualize these with some volcano plots, and I have a few volcano plots that I'll show you here shortly. So let me just start with these questions about how well do they correlate with internal dose. That's a question we receive often. So we've done several different pilot scale studies to look at correlations with internal dose. The publications are all listed here. In all of these studies, we had some of these are children, some of these are adults, anywhere from 30 to 7 people wear a wristband for 5 days. And we would ask those participants to collect their first morning void urine samples on days 1, 3, and 5, and we pooled them. And that pooled urine sample was analyzed for the standard biomarker of exposure, the specific metabolite for each of these compounds. So on the left, you see chlorinated tris, or TDCPP, was well correlated with its metabolite in urine, BDCPP. In the middle, that's benzyl butyl phthalate, right? A phthalate that's common in things like vinyl flooring and other plastic products. And on the right is DEET, the insecticide. I know that Dr. Anderson mentioned. This was one that was really surprising to me. Not only was it a very strong correlation, you can see here of 0.92, that data actually came from our non-targeted analysis and not our targeted analysis. So what you're looking at there on the x-axis are actually the normalized area counts. So it's a semi-quant method, but it was nicely correlated with the urinary metabolite, which is a targeted analysis that was performed by Antonia Califat's group at the CDC. So that was really exciting for us that we could see that even the non-targeted data is providing reliable, robust measurements for some of these chemicals. Now, these are all chemicals with short half-lives. We wanted to look at chemicals that had long half-lives. So we did some other studies with brominated flame retardants, and namely PBDEs. The one on the left is from adults, the one on the right is from children. So you can see that BD47 has a half-life of about 1.8 years. This is what it's estimated at. It was correlated with blood serum levels of BD47 in this pilot study we conducted. These are samples collected in 2016. It was correlation coefficient of about 0.52. There's definitely some variability there, but given how long this chemical is in the blood, it was really nice to see that just wearing it for, in this case, that was 7 days, did correlate with the blood levels. Probably because most of our exposure to these compounds does occur in the indoor environment, and most of these people did live in the same home for the last couple of years. And then on the right, you have BD209, which is estimated to have a half-life of about 2 weeks. This was a smaller number of children ages 4 to 6 that wore this, but we did have a really nice correlation. So this was really exciting for us. And this is where I said, I wonder if this is capturing chemicals that are exposures through inadvertent dust ingestion, because BD209 is not really volatile. It's really associated with dust and particulate phase, given how hydrophobic it is. And it is thought that for BD209, that most of our exposure, particularly in children, is through hand to mouth or inadvertent dust ingestion, but we did have a nice correlation. We're hoping to see if we can repeat this in the future. And then we get this question about PFAS a lot, because I know PFAS is a big topic. For PFOA and PFAS and PFHXS, these common PFAS that are of concern for cancer and health risks, we know that diet is a main exposure route for those chemicals. So I don't think wristbands are useful for those main perfluoroalkyl acids. However, there are some PFAS that are used in building materials. Think about your stain repellent carpets and upholstery and even paints. And many of these are what we call PFAS precursors because they can break down to the perfluoroalkyl acids like PFOA. In a recent study we conducted, we did find a very nice correlation between methylphoce, which is a volatile PFAS on the wristband, with N-methylphosa in the blood. And this is a known parent metabolite mixture. So methylphosate is oxidized to N-methylphosa. N-methylphosa is one of those 7 PFAS that are recommended for monitoring for clinical guidance by the National Academies report. And while so most PFAS, I do think guide is exposure. I do think in this one case, N-methylphosa, it seems our data seems to suggest that exposure is likely inhalation or dermal exposure and coming from the indoor environment. Now, this was a population in Michigan. We have since repeated this trend in a population in North Carolina. That data is not published, but it was statistically significant. So this seems to be similar in other areas. Now, not every chemical is correlated with the metabolites. All right, so this is from one of our pilot studies where we were looking at pesticides, and we were able to detect chlorpyrifos on the wristbands, 83% detection frequency, and 100% in the urine. And those samples are analyzed by Antonia Califat's group at the CDC. But we did not see a significant correlation. And I think this is likely because there's low-level residues on our food, and diet's just more important. So we don't see that correlation. But that doesn't mean the wristband measurements are invalid. It's probably just picking up this low-level exposure in the home. And I particularly say that because we have a collaboration with a group at Johns Hopkins University where they were deploying wristbands in Central America in an area where they were applying chlorpyrifos in the fields. And we analyzed their wristbands. And first of all, the chlorpyrifos measurements on their wristbands were much higher than what we've seen here. And they were significantly correlated with this same urinary metabolite in their cohort. And they presented on that at the ISCS meeting. So I know they're working on that, and that's in draft and should be published soon. So there's going to be some cases where you don't see a correlation because exposure is from the diet, but in other populations, when it's a non-dietary exposure, you could see that correlation. Then we get the question to you about, can you use these in children? I think just to, like Kim Anderson suggested, we've used these down to age of 3 in children. But then there's this question about infants, right? They're a vulnerable population, very difficult to get blood and urine from them. So we tried this use of wristband, but made it a little bit bigger and put it on the ankle. So an ankle band. So we conducted a pilot study with 21 infants here in North Carolina, where we had them wear one of these ankle bands for 3 days. The kids were between 6 to 18 months of age. And here we just asked the parent to collect a spot urine sample sometime during that 3-day window. So either using one of these pediatric urine bags or one of these toddler potty training units, and then transfer it to a urine specimen cup. And these were all analyzed in my laboratory. Here's some of the data. This publication was just accepted last week, so it'll be out soon in ES&T letters. But we can see for the 2 main organophosphate esters that we're able to measure, particularly in urine in our laboratory, we saw very nice correlations that were statistically significant. On the right is TDCPP, that chlorinated organophosphate flame retardant. On the left, what you're looking at is the diphenyl phosphate metabolite in urine. And on the x-axis is the sum of all the parent molecules of DPHP. Our panel has 30 different organophosphate esters in there that we target and measure. So we looked at the sum of these because all of these could potentially break down to diphenyl phosphate, and there was a significant correlation. This also just highlights another advantage of the wristbands because sometimes with biomonitoring, you're looking at a metabolite that has multiple parent compounds, right? So you can't always know which parent it came from. But using the wristbands, you're getting information on the differences in exposure for the various parents. So that's just one nice advantage about using these wearable technologies. I still heard someone ask about the variability in measurements around the wristband. We have looked at this as well. I know Dr. Anderson has as well. In our study, we said, well, what's the difference if we take a sample here versus a sample here versus a sample here? This is published in our paper from 2022. It's in the supporting information. So we took 10 wristbands and cut 3 pieces from 3 different sections and then calculated the % relative standard deviation. We only focused on chemicals that had 100% detection in every single sample, so we can avoid any biases from imputation. But you can see here for a wide range of organophosphate esters, brominated flame retardants, PAHs, pesticides, and phthalates, that % RSD was pretty low among those 3 different samples around the wristband. I've highlighted the 20% mark because that's a typical threshold in many EPA methods. So you can see almost everything was below that. There was a few that were higher. There was some variability, but overall average was about 13%, which is actually pretty similar to the precision measurements you get in a lot of methods using mass spectrometry. So we felt really good about this. How do they compare to spot urine? I know that this question was asked too. We did do one small pilot project a few years ago where we asked people, 10 people, to wear 5 wristbands and take one off every day. We also asked them to collect all of their urine, a 24 urine sample, each of those 5 days. So we gave them a very large jug, and we did compensate them because I know this is not fun. We also asked them to collect a spot urine sample at different times on those 5 days. So using the 24-hour urine samples, we can look at total mass excreted over those 5 days. And then with these different wristband measurements, we could look at the concentrations on those wristbands over those 5 days, depending on if they wear it 1 day, 2 days, or 5 days. So what I'm showing you here is actual data for one of our participants for the flame retardant TDCPP. So you can see there was variability in the urine every day, and then this rather linear uptake on the wristband. And then we just compared these, right? Spot urine as a predictor versus wristband as a predictor. So the top panels are data for TDCPP, and the bottom panels are data for TCPP. Those are 2 different chlorinated organophosphates. The left is the correlation between wristband and total mass excreted. On the right is a spot urine sample versus total mass excreted. Now, when we picked spot urine samples, we had 5 different spot urine samples. So you get a very different correlation depending on which day you pick. The right I'm showing you here is just day 3. It was in the middle. So for TDCPP, the correlation for spot urine was a little bit higher than the wristband, but for TCPP, it was lower. But what you see if you use a different spot urine sample, sometimes it's significant, sometimes it's not. And it's 10 people, so we have to take this with some caution. But if you look at the spread of the data with the wristbands, to me, that seems a little more robust and linear compared to the variability you see on the right. Now, these correlations are Spearman correlations, which are rank sums, so they're not looking at linear relationships. But this data is also published. And then we took those samples and reanalyzed them for phenolic compounds, namely parabens and triclosan. And that is also published in the Levasseur 2024 paper. And we actually saw something very similar. Lots of variability depending on what spot urine you pick, but the wristbands actually predicted it, total mass excreted fairly well. So they're at least as good as a spot urine, I would say sometimes better. Now, we couldn't analyze every chemical in our panel in urine, but we did have data in all these different chemicals in the wristbands. This was published in the Salmon et al. 2024 paper. I'm just showing you 3 chemicals I picked out randomly: diethyl phthalate, phenanthrene, and permethrin. Each line represents a person in the study and what their levels were, depending on if they wore it 5 days, 3 days, 1 day, etc. Right, so the slope of that line is the difference in their exposure. And you can see for most of these, it looks like a nice linear uptake. I will note that there's very different Y scales here on these different chemicals. For example, permethrin is on a log scale. Some folks had 1,000 nanograms per gram of transpermethrin, and others had 10. We think a lot of this is actually due to use of PETs, because that's a really common flea and tick medication in pets. So we often see people with higher levels are pet owners. But you can see all those, so that this is a nice measurement. Now, there's some variability. Some people had higher levels on day 1 versus 3, but overall, most people, you see this nice linear uptake for many of these chemicals. So now I wanted to kind of just switch gears and talk about how we've been using these with firefighters, because it just as an example of the type of questions we're asking with wristbands. I'll start with this paper that was published in 2019. We started collaborating with the Durham firefighters here in North Carolina, and we wanted to ask this question about how different their chemical exposures were off duty versus on duty, and even how they were different when they responded to a fire or not. So this was a study that was led by my former PhD student, Jessica Levasseur. And so we had more than 20, but we ended up giving multiple kits to firefighters and just kept asking them to wear one until they completed a wristband or wore a wristband for all 3 periods here, either off duty or home for 6 days, or working with or without responding to a fire. And so when those samples were analyzed, we performed some regression analysis on the data. So these are the targeted data results. So what you're looking at here is changes on duty versus off duty. So the horizontal line at 1, that's kind of a baseline or the average for all the firefighters, their exposures off duty. And then the y-axis is a multiplicative change in their exposure on duty compared to off duty. So you can see all of these symbols are above the one line. So their exposures were higher on duty versus off duty, which is what you'd expect for these, which are PAHs, combustion byproducts. Many of these were statistically significant, which is whether it's filled in or not. And you can see the squares are higher than the circles, right? So their exposures were higher when responding to a fire, which makes sense. That's what we would expect. What was interesting to me is, why are they also higher if they're working but not responding to a fire? So it seems like they had additional pH exposures even if they didn't respond to a fire. So is that because they went to a training event and they didn't tell us that was a fire event? Or is it because they respond to a car accident and there's some exposures, so it's not fire but it's also work related? Or is it because there's exposure in the station or through the rigs? We just don't know. But to us, this is an important question to ask so we can understand where these exposures are coming from, because particularly in firefighters who are worried about cancer risk, we need to do everything we can to mitigate those exposures. And we use the same approach for brominated flame retardants right here, looking at exposures on duty versus off duty. So like the PAHs, the BFRs are also higher on duty versus off duty, many of them statistically significant. Some of these are legacy BFRs that have been phased out, and some of these are current use alternative BFRs. But what you can see is here, there's not much of a difference between the circles and the squares, which means it doesn't seem to be related to a fire event. It's just something about being a firefighter overall that led to higher exposures. So my question is, why do they have these higher exposures when they're working if it's not related to the fire? And I don't think we know the answer to that yet. Is it something about the equipment they use? Is it something in the fire stations, the rigs? We're not quite sure. And it is one study with 20 firefighters. And we are actually working more with the firefighters to answer some of these questions, but I think that's an interesting one. And then you'll get something like phthalates, and phthalates actually were generally not different on duty versus off duty, with the exception of a few of these low molecular weight phthalates, which are actually higher off duty than on duty. We hypothesize this is due to use of personal care products, that they use personal care products when they're off duty or home that they weren't using when they were on duty, because we saw something similar for some pesticides that are common indoors. And then we switch to our non-targeted analysis of data. This is with the same fire department, although this was a second pilot study. So these are samples that were collected in 2023. These are not yet published, but I wanted to give you an example of some of the work we can do with wristbands using a non-targeted approach. So here you're looking at a volcano plot where every dot on this volcano plot is a different feature or a different chemical that we picked up in our analysis, right? The x-axis is the fold change in the relative amount of that chemical on the wristband. And here we're differentiating between wristbands that went to a fire and those that didn't go to a fire, right? And the y-axis is the p-value. And so what's statistically significant is in the upper right-hand corner, we had 22 features that are not in our targeted panel. One targeted chemical was statistically significant, and that were higher in these wristbands that went to a fire. But you can look at the volcano plot and see there's a skew to the right. So overall, there's more exposures that are ongoing within firefighters. Some of these were statistically significant, some of them were not, when you're responding to a fire. And that makes sense, right? This is only 23 people, so it's still a small number. We also asked the firefighters to record the total amount of time they were at a fire. So if we plot that, we just here plotted by the median. So the median amount of time a wristband was at a fire in this case was 0.75 hours or 45 minutes. Well, now it's more statistically significant, right? Because you're really separating things that spent longer closer to a fire. Probably makes sense. Now we have 75 unknown features and 4 targeted features that were statistically significant. And again, you see that skew to the right. More time at a fire, more exposures overall. But what really I found fascinating, which really confused us for a while, is this one where we plotted years as a firefighter. We had quite a range, and 23 firefighters. Some were right out of the academy, only had been working here 2 years, and then we had other firefighters that had been there for 30 years. The median was 15.5 years. So here we're splitting at the median, and what you see is that there's much more exposures in the participants that had been a firefighter for fewer years, as opposed to more years, which is not what we expected. Now we've highlighted a bunch of flame retardants in here because I took this slide from one of our talks, but here we had 500 features that were statistically significant. So this was baffling to us until we started talking to our fire service partners and learning more about what they're doing. And what we learned is that it's often the new recruits that are the ones going into the fire, performing the overhaul, more active on the scene. And it's the fire chiefs and the battalion chiefs that are coordinating farther away from the fire. So we think this may be due to the different tasks they're performing based on their seniority in the fire service. Or maybe it's just that younger people are more active and they have more exposures to things. It is a pilot study and needs more, needs replication, needs to be looked at some more. But if this is true, it's an interesting thought, because that means maybe some of the exposures that contribute to the risk for cancer are happening when firefighters are first on the job, maybe over the first 10 years. But I think it's something, again, that needs to be looked at. And this provides an example, again, of some of the differences we're seeing in exposure patterns, and how you can use the wristbands to kind of glean this type of an insight. So I'm almost done here. I just wanted to I know there's a question came up of how you use these in exposure framework. I know there's a question. Can you take them and estimate indoor air concentrations? And I know that's a direction a lot of people are going, and I've seen that happen. I think when they're stationary, I personally think that's okay. I know there's been a couple of calibration studies that have already been published. I'm more thinking that maybe we should be thinking about how we can use the data to predict internal dose using machine learning algorithms and having lots of training sets, because the data seems to suggest it's integrating both thermal and inhalation exposure. But on top of that, I worry about movement, right? Because what's happening is this is a flux calculation. Flux are influenced by movement. And I wanted to evaluate this. So I collaborated with my colleague here in the Civil and Environmental Engineering, Professor Helen Shi Kim, and her PhD student, Josh Miller. And what we did is we took these wristbands and we put them on a test tube rotator, and we deployed these in the family room of a house here in North Carolina for 30 days. And we had multiple of these test tube rotators with the wristbands going at different speeds. And so we estimated the speed. You can kind of see in this diagram to the right how we measured the dimensions, and then they rotated and they didn't touch anything. They're only touching air, but they rotated at different speeds. And if you look at our data, this was accepted for publication just recently, you can see that speed has a big impact on accumulation rates. So these are three different chemicals. There's tris-chloropropylphosphate, there's 4-ter-octylphenol in the middle, because we picked that up quite a bit, and liliol from personal care products on the right. Now, first you see that some of these look linear and some look like they're reaching an equilibrium. So it is important to figure out where those occur. But what you can see is if it's stationary, which is the black line, versus moving at 1.1 meters per second, which is the green line, there's a very big difference in the rate at which these accumulate. So 1.1 meters per second is basically a walking speed, right? So if you're stationary versus walking, the rate at which that accumulates on the wristband could be very different, right? And so kind of using this, we estimated what we call an enrichment factor compared to worn wristbands and looked at these relationships, and there was one with KOA. The point being that I think the data suggests that what accumulates on the wristband is a very complex process. It's not solely driven by gas phase partitioning. There's the role of the skin, the dermal exposure, and particularly maybe particles. Now, we don't rinse particles off of our wristbands. We do keep them on here, and we've seen some correlations with chemicals that are particle bound. So I think it's just multifaceted, and there's a lot yet to unpackage on what pathways are contributing to accumulation on the wristbands. So advantages, I think there's many. I know we've already discussed them, right? They're non-invasive. We can use these to measure exposure to hundreds of chemicals over time. You can mail these back and forth. You don't have to have a clinic visit. I like looking at patterns of exposure, and I'm really excited to use them in those applications. And lastly, these can help us reach some, isolated communities that may have difficulty participating in research otherwise. Now, limitations, as Dr. Anderson already said, right? They don't work well for metals. I get worried about their use for chemicals that have higher water solubility, like glyphosate, or very, very volatile, because we just have a tendency to store them for longer periods of time, and I worry about that. And I think it is, I know we mentioned hand dominance, so I was really glad Kim mentioned that. I think we need to understand that a little bit better for the general population. I still don't know what the difference is if you wear long sleeves versus short sleeves. That's another factor. Physical activity, I think, has to be looked at. And I think we need to understand more what time points, where do you reach that equilibrium? All of our studies are usually either 5 days or 7 days, like Dr. Anderson, just trying to keep them. It's also logistically easier. Now, our lab continues to support some of these other ongoing studies. We are working with the firefighter cancer cohort study. We also are working with the NIH Cure Consortium, which looks at chronic kidney disease. So they have wristbands deployed in 6 different countries. We're analyzing those in our laboratory right now. And we're supporting wristbands for 2 of the 5 seeker cohorts, which is an NCI-funded study to look at environmental exposures and cancer risks. So those are all ongoing. Certainly want to thank my lab groups that's changed a bit over the time, and all of our collaborators and funders. And I'll stop there. So thank you for letting me go over time a little bit. Thanks.
Thank you so much. Tom, go ahead. We'll do 10, 15 minutes of questions, and we'll take a quick break before we discuss more.
Tom McCone. Thanks, Heather. That was very interesting. I want to start with just 2 quick comments. One is you were talking about chlorpyrifos, and in an agricultural community, you expected more correlation. I think about a decade ago when we were looking at organophosphates in the Salinas Valley, we found out that the biomarker levels in the women were higher compared to NHANES, and they were systematically higher, reflecting that the indoor environment there was equilibrating and accumulate PAHs, and that made up the difference. So we actually saw that in Salinas, where if it's used locally, your home or your living environment will trap the organophosphates and serve as a nice delivery system to the population. So that's consistent. Another quick comment is, I have a friend who's a firefighter. They spend a lot of time driving around in their diesel trucks. So I don't know if you thought about that as the added source for PAHs. You know, if they're not
That's
fighting
exactly a
fires.
question we want to answer.
Yeah, they're always out, buying groceries or just driving around they don't sit in the station all day. And even if they do, they run the trucks half the time and get exposed. But the question I have, and this is sort of a broad question that we may discuss later, is, you've really taken us in the direction of showing, how we're moving more and more to using wristbands to be more quantitative, to characterize either concentrations or biomarker levels. But if you could speculate where we are now in having wristbands as a key element of an exposome. I mean, are we starting to get there? How many years? I mean, have you thought about what it might take to actually make this a piece of what we call like the exposome, the record of what people are exposed to, and a reliable one? And again, I think it's moving there, but you might have a better sense of if it's likely to move in that direction.
That's a great question. I do know that some of the larger exposomics initiatives in the European Union are including wristbands in many of their studies, and they're moving ahead with those. NIEHS just funded, one center for exposomics here in the United States. And my understanding is that they are trying to do some lab calibrations or harmonization studies to think more about how wristbands can be used in these exposomic studies. So I do think we're going to see more of those moving forward. Yes. My personal thought is we can't replace blood sampling with the wristbands, but I think it's an important complement to have, because there's always limitations of blood sampling and urine sampling, right? But I think we need both of them to understand the full picture. And personally, I'd love to see us use them to understand exposures and how they change over time. I get really worried about cross-sectional analyses or just measuring them once a year. I didn't have time to get into it, but we have another study where we looked at variability by latitude and longitude and temperatures and over seasons, and you just see very drastic exposure profiles that change. So I just think there's a lot of questions that could be answered or low-hanging fruit by using the wristbands.
Hi, Ulrika Luderer. Thank you. That was a really great presentation. I just have a question, related to the firefighters where, when they were on duty, but there was no fire, the levels were actually kind of paradoxically higher, right? And so I was wondering whether you have or are planning on looking at things like turnout gear and levels of, PAHs and, other chemicals on the turnout gear because as I understand it, they're not always being cleaned all the time, from one fire to the other. So just a question about
Yep.
that.
We have done that. We have a paper under review that's looked at both PFAS and BFRs in turnout gear. And I share your concern that there could be some exposures that are just coming from contaminated gear. Yes. The topic of gear within the fire service is very controversial right now because of the PFAS issue. So it becomes really complicated. But yes, I do think that's a factor that and something that needs to be looked further into.
Oh, Heather, it's Kathleen Atfield from Biomonitoring California. Thank you so much for talking about the issues about the interpretation of thinking about inhalation versus dermal is, what does it represent? Because that's been something that's always confused me, to what extent is this telling you what it's absorbing from the air versus what might land on it from a pesticide spray or use of a personal care product. But since you already started to address that, I'll ask something sort of similar. What about from sweat or from sloughing of skin, like things actually coming off the body and into the wristband? Is that something you've thought about, and how do you talk about it?
Yeah, I do get the sense that wristbands are picking up dermal exposures. So I do believe that there are chemicals that are in our air. Some of them absorb to the wristbands, but some of them just absorb directly to our skin, right? And so what we pick up on the wristband could be coming from these chemicals that are on the skin onto the wristband. Certainly we see things like squalene and other biological molecules that we know the skin secretes on the wristband. Now, what fraction of that is from dermal versus inhalation is very difficult to tease out. I know of one paper that tried to look at this. That was a group at Indiana University, and I think their data suggests that both routes of exposure are integrated in a wristband measurement, but it's going to be different for each chemical, and it's hard to tease out. I think this is an area where we do need some more research to understand this a little bit better. I know we're often asked the question, like, do PBDEs in our bloodstream, are we excreting them out our pores and that's accumulating and that's why we see a correlation? I don't believe that is happening. I think it's more of that we're picking up this signal from the air or the particles in the air on the wristbands, and sometimes those stick to the skin and they are on the skin and then they partition onto the wristband. Not that we're excreting it out, because I just think that the thermodynamics of that, and I am not a dermal absorption expert, I just think the thermodynamics of that would be very difficult. So to me, the more likely explanation is that our skin absorbs these chemicals as well, and then there's some transfer to the wristband.
Thank you, Martha Sandy from OEA and Biomonitoring California. So along those same lines, I was thinking, could someone be excreting something in the sweat and it's absorbed to the wristband? And I guess you could look in the wristband for a metabolite that we know is excreted in the sweat and see if you pick it up in the wristband. That's an experiment that could be done, but are you aware of anyone doing that?
Not. Certainly we could ask Dr. Anderson if she's aware of anything. I haven't come across a lot of studies where they actually identify metabolites of chemicals coming out in sweat either. I know this has come up a lot in the firefighters because we get this question a lot of whether they can go in a sauna and they'll sweat them out. And I know there's some evidence to suggest that some of the PAHs are reduced on the skin from sauna, but I'm wondering if those are just ones that were on the skin to begin with and were not fully absorbed. I just don't think we know. But I think that's an excellent question and something that should be explored, yes.
And a related question, you did say you're wondering about long sleeve versus short sleeve. So we know that some clothing is treated with PFASs and maybe, a variety of other compounds. Have you again tried to do some sort of a controlled experiment with wristbands and people that are wearing clothing, has something and it's in contact with the wristband versus people that aren't? And have you ever done that type of an experiment? Or I guess this question could also be for Kim later.
We have not. It's certainly been on the back of our mind, and it's on kind of these list of experiments we would love to run if we have enough time. And we started off really trying to understand more about these relationships between wristband and internal dose. So multiple studies there, and then the 24-hour urine study. So we just haven't had time, and we don't always have funding kind of to do all these additional validation studies. But I agree, it's an important question to ask.
And we do have one public comment too. We need to get
I
to.
was just going to ask about that.
Kathleen Atfield again. Same question I asked of Kim, sort of what are the missteps you see people making in trying to use silicone wristbands, and what would you advise us to look out for?
I haven't seen a lot of missteps. I mean, most people are happy to wear them. People often forget to mail them back. We have to remind them. In our case, we do normalize to deployment period. If somebody wears them for 5 days and somebody else wears them for 2 days, we normalize to per day. Just based on our studies, it seems to be fairly linear for us. So we feel comfortable doing that before we apply statistical methods to analyzing the data. But as I said, there's more questions that we have to answer. Like, is there variability from the clothing, hand dominance? I get nervous about measuring things that are too volatile because they'll wear it, then they hold on to it, then they'll mail it to us. And I don't know how long it's been around at 25 to 30 degrees Celsius for some of those really volatile chemicals. We stick to chemicals where they are a certain range. I know Dr. Anderson mentioned this, right, of KOW or KOA, where we feel more confident that the chemicals are more stable on the wristbands over time and don't go into the more volatile things. I just think what we have to be aware of is there are different methods being used to analyze these. And so that can contribute to differences in what you detect or don't detect, just because the methodologies are different.
Rebecca, do you want to invite the commenter online?
Yes, and this comment was submitted by Lily Wu, and I'll read the comment. It says, thanks for your presentation. Along the lines of other silicone passive samplers, such as infant ankle band, there are silicone brooches that could be worn closer to people's necks to potentially get a better indication of inhalation exposures. Do you know of any studies that might compare a wristband versus a brooch type wearable?
Yeah, so I do know that Dr. Miriam Diamond and Marta Venier, Miriam Diamond's at the University of Toronto, Marta's at Indiana University. They've been looking at that and they have a paper out. I can't remember what the exact differences were off the top of my head, and sometimes it's restricted to like one class of compounds. But there is some data out there on that I get worried about using a brooch. I don't think there's anything wrong with it. I think it will work. I just want our participants to wear it for more than a day, right? And you're changing clothes. So for us, the ideal situation is to have someone put it on and forget about it. So you don't change your behavior, you don't do anything differently. So the wristbands are nice in that regard because you can put them on, go about your daily behaviors, activities, and you don't have to worry about it. But a brooch, you're gonna have to take on and take off when you change your clothes. And I worry just a little bit about contaminating it in that way.
We'll do one more question and then take a little break.
Ulrika Luderer again. I just have a couple of really basic questions. So one of them is, I mean, have you looked at all whether people, with bathing or washing, whether that makes a difference? Are you losing things from the wristband? And also, how long, have you looked at various different times, that you've stored the wristbands to see whether the things that you find in them change over time, from the same wristband, for example, do that kind of a study?
Right. We have not done the latter yet. I agree that's very important. With regards to showering and bathing, when we conduct all these pilot studies, we ask that question: how often do you bathe or shower or go swimming? When we looked at the survey data, we don't see any difference. That may be because most people in our pilot studies are showering every day. There's not a ton of variability there, but even the data I presented to you where we started off with 5 wristbands and we took one off every day, all 10 of those participants were like showering just about every day, and you still see a linear uptake with time. So they're not being washed off. And the chemicals we focus on, again, are very hydrophobic. They are not very soluble in water. So I don't think it's very likely that they're going to wash off during showering or in the pool or things like that.
Great. Well, thank you for an excellent presentation. We'll go ahead and take maybe a 10-minute break.
Yeah, 10 minutes and we can come back at 2:50.
2:50. Okay.